What if we could cut the cost of life-saving therapies by 90%? Imagine a world where a 2 million treatment becomes $200,000. Still expensive, but suddenly within reach for many more patients. This isn't just wishful thinking. Right now, brilliant minds across the biotech industry are revolutionizing how we manufacture these therapies, and their innovations are already showing promising results. Welcome back to the Smart Biotech Scientist podcast. I'm your host, David Brühlmann, and throughout my career, I have seen how manufacturing innovation can dramatically reduce costs and increase access to life-saving treatments. Today, we are at a turning point where new approaches to bioprocessing could transform not just how we make therapies, but who can access them. If you haven't listened to parts 1 and 2 of this 4-part series yet, I encourage you to start there first, as they lay important groundwork for today's discussion. In part 3 of our special series on the evolution and future of bioprocessing, we'll explore how industry leaders are tackling the 5 critical challenges we had discussed last time. From groundbreaking process designs to revolutionary quality control approaches. These innovations aren't just technical achievements, they are pathways to making these treatments accessible to millions more patients worldwide. And it's not just about therapies. These same innovations are helping make sustainable, cultivated food a reality for our growing world. Let's dive into the fascinating solutions that are reshaping the future of bioprocessing. So let's look at how industry leaders are addressing these challenges to enable widespread access. The first one is the process itself. So how can we revolutionize the bioprocess design? When I was working at Merck, we put a lot of effort into enhancing the antibody production. And if you're familiar with antibody production, we have a certain number of seeds, train reactors in a batch mode to ramp up the volume. And then once you have enough cell, you seed your production bioreactor. And that's typically run in a fat batch mode. Instead of doing the last seed train called N-1 in a batch mode, we switched to a perfusion mode reaction. And this has several advantages, and one of them is that you can extend the culture and you can reach much higher cell densities. And that's great because if we can reach more cells in the N-1 stage, we can start the production stage with a lot more cells. And inspired by this work, ICNOS, they developed a intensified process to produce a difficult to express multispecific antibody. And they first started by obviously optimizing the cell line itself. And once they had a suitable cell line, they went into the fed batch process first. And like a typical fed batch process, you reach about 20 million viable cells per mil. And they reached about a titer of 6 grams per liter for this difficult to express protein, which is already very good. Now moving into a high density process, they were able to reach 70 million viable cells per mil. And because of that, they were able to reach titers of about 10 grams per liter. And now for clone C, they also tested the very same process where they were able to reach even a higher titer of 11 grams per liter. Again, for a difficult to express protein. Moving back to the cell and gene therapy space, Oreo Biotech, they are leveraging an integrated approach. They're combining biology, engineering, and the digital, and their bioreactor looks like that. And they leveraged all the knowledge we acquired in the biologic space and transposed it to the very specific challenges in the cell and gene therapy space. It's a fully closed system. It's an automatized system, enabling small batch sizes and very small in different sizes, very flexible. And they work to automatize the longest and most manually intensive part of this process, which is the activation, the transduction, the expansion, and the harvest. And this not only increases the efficiency, it also increases finally the outcomes of the process because it's less manual. It's automatized. It's much more robust and repeatable because it doesn't depend on an operator variation. So they were able to get a 2.5x higher cell growth just by changing the system. On the downstream side, we are facing yet another challenge because we are moving away from traditional antibody formats. We are now seeing proteins that lack the Fc part of the molecule. And if you know something about proteinase, specifically targets the Fc part of the molecule. And Cytiva recognized this challenge and they developed a new affinity chromatography resin that also enables high purities, but now for molecules lacking the Fc part. The way they do that is when you develop your protein of interest, a sequence, you add an additional amino acid sequence to it, and then a tag is also expressed at the end of the protein on one of the terminus of the protein. Now, when you load this protein of interest onto your column, again with a traditional antibody, the ligand specifically recognizes the protein, it specifically binds to it, and now comes the genius part. When you elute, not only is the protein of interest released, the tag actually stays on the ligand. So only the protein of interest is released, and that solves a lot of problems, a lot of regulatory problems.
Now let's take a short break and shift our focus. We have seen how innovative process designs are transforming manufacturing efficiency. But efficiency alone isn't enough. We need to ensure consistent quality at every step. As we continue, we'll explore how industry leaders are revolutionizing quality control in ways that could make these treatments not just more accessible, but more reliable too.
The second part industry leaders are on a mission is in the quality by design space. And this is very critical, especially in the cell and gene therapy space. And one of the companies leading the space is Cytofacto out of Kobe, Japan. And they specialize in the production of stem cells. And now cells, again, imagine this complexity, much higher complexity of cells. Cells are so much more complex that they react to the slightest differences of shear stress, of temperature, of pH, of media components, raw material lot difference, and so on. And this is a big problem that oftentimes we don't handle them as well as we should, and then we lose potency or viability or all kinds of things. So we cannot treat as many patients as actually we should be treating. And one more thing is that these processes cannot undergo traditional process validation. Because if you have a patient population of 1, you cannot validate your process. The same challenge for stem cells, and you have a lot of differences between one cell type and another. So the way they're solving this is by, again, leveraging an integrated approach. It's a fully closed system, fully automatized, and on top of that, they have a sophisticated PAT system. So they do the seeding, the culturing, passaging, and harvest, everything in one system combined. And because they have a sophisticated PAT system, they are able to monitor all the critical process parameter and also obviously the critical quality attributes in real time, such as media components, such as intermediates, such as growth rate, morphology, and even clinical outcomes. Also on the cell and gene therapy, recent work that has been published, it's a novel refractory-based PAT system. It was developed by a team of Oxford Medica, UCL, ETH, and What B Innovation Limited. And what they did is they're using a PAT system in a bioreactor and the first time for a lentivirus production process in HEC293T. And they were able to measure in real time, monitor in real time the metabolic activity, not just how many cells you have, but really the metabolic activity. And they built a model of more than 350 reactions and about 335 metabolites, which is quite a complex model. And they were able to create that and sophisticate the model so that this model is sufficient to collect data for a routine operation, which shows that this has a huge potential, not only in more established bioprocessing or even in chemistry, obviously has been around for a long time, but also in the specific challenges of the gene therapy. And then you can take this a step further. All the sensors you put on your system, for instance, your bioreactor or your column, you link them to a virtual bioreactor. And using AI, you can predict process outcomes, you can monitor your run, you can do all kinds of fancy stuff. So Sanofi took that and integrated everything into one single thing, into their digital factory that is now 80 times more productive. It's a very combined approach with internal realization. They connected team operations. They have the equipment and plants, real-time data-driven, and so smart quality. You see the potential where this goes. Also in the quality side, but a slightly different note was this. One day when I was working in the lab, I came across this intriguing article. It read, "Influence of pistachios on performance and exercise-induced inflammation, oxidative stress, immune dysfunction, and metabolite shifts in cyclists." And I thought, metabolite shifts, that sounds interesting. Let's look that up. And as I was reading through the paper, I came across a list where they listed the various components of the pistachio nut that induced a metabolite shift in cyclists. Then I said, wait a minute, if this works in cyclists, could it also work in CHO cells? And long story short, yes, it works also in CHO cells. And one of the components of this list was raffinose. And raffinose is a trisaccharide, so a sugar molecule. And we were able to supplement cell culture media to influence the glycosylation profile of an antibody. So I don't know if you're all familiar with glycosylation. Glycosylation is the sugar molecule on the antibody, and it has an impact on the efficacy of your molecule and also the stability. So we were able, by just adding one single sugar, to specifically target one specific, so the high mannose glycan forms specifically. That's genius, not by changing the cell line, just by adding something in the cell culture media. And then we thought, well, we could actually engineer the quality of our molecules and make them more efficacious. That's what we tested with a much more complex molecule, a fusion antibody with 3 distinct glycan sites. And again, by just supplementing the media with various components, we were able to increase the receptor affinity. And then ultimately, we were able to increase the relative ADCC reported potency, which means we were able to enhance the efficacy of our fusion antibody by just inducing a metabolite shift in the cell.
That concludes part 3 of our 4-part series on the evolution and future of bioprocessing. We've explored how industry leaders are innovating in process design and quality control to make breakthrough therapies more accessible. These aren't just incremental improvements. They are fundamental shifts in how we approach bioprocessing. But this is just the beginning. In our final episode, we'll dive into the disruptive innovations that could completely transform how we manufacture biologics and cultivated food. We're talking about technologies that seemed like science fiction just a few years ago. Are you ready to accelerate your bioprocess development journey? Book a free 30-minute assessment call to explore how we can help you overcome technical hurdles and optimize your manufacturing strategy. During this focused consultation, we'll identify your key process development challenges, discuss potential optimization opportunities, and outline strategic approaches to reduce costs and timelines. As we have explored in this episode, innovative manufacturing approaches can dramatically reduce therapy costs and improve accessibility. Let's discuss how these innovations could benefit your development program. Visit BrühlmannConsulting.com to schedule your complimentary session. Together, we can work toward making life-saving therapies more accessible. And join us next time for the exciting conclusion of our 4-part series. This is David Brühlmann, and you are listening to the Smart Biotech Scientist podcast. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast.
Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.